Good afternoon, children. Welcome back to the lecture series for Zoology First Year Intermediate. I am Vishnu Priya, Principal, TSMS Palma Class. Today we will begin another interesting chapter, Animal Diversity 1. What makes our planet unique is that only on Earth life is possible. The life forms on Earth are highly diverse with many kinds of organisms. Of the 6 kingdoms of life, the animal kingdom or metazoa is one of the most fascinating. The diversity of this kingdom is so vast, consisting of organisms like us, the human beings, and the mayfly, which has a shortest lifespan, and the immortal jellyfish, which never die. An animal called the cyphalophore was recently found in the ocean, which is longest animal living. It is about 180 feet long. These organisms are very intriguing to study. An interesting fact is that earthworms are very flexible and elastic, unlike humans. This is because they do not have a notochord or the backbone. Such organisms without notochord are called invertebrates. Invertebrates are the largest group within the animal kingdom. In addition to not having a notochord, they are multicellular and reproduce so quickly. Now, I would like to introduce you to our esteemed speaker of today, Mr. Shankar Naik, FSC Principal, TSMS Imampet, Suryapet District. He is a winner of the Best Teacher Award at the district level this year. He is also an excellent and experienced MSET coach and TSPSC competitive exam trainer. And now, Shankar Naik sir will give you a deeper understanding of invertebrates, their further classification into phyla and classes, and about the special characteristics of the invertebrates. Shankar Naik sir, you may please go ahead.
Thank you, madam. Thank you so much. Thanks for happy and kind words to me. Kind words about me. Thank you so much, madam. Thanks to, I would like to say special thanks to Additional Director Garu and Joint Director Garu, along with Manjari madam, Vijayalakshmi madam, and Vishnu Priya madam. Thank you so much. Thanks for support and thanks to make me to be here in front of intermediate first year students. Thank you, one and all. Thanks for giving this opportunity to me. Right now, I'm going to upload my slides. I'm sharing right now. Okay. Once again, good morning, students. Today, we are going to discuss Animal Diversity 1. Animal Diversity 1 is a third unit in intermediate first year zoology. So, you know what is the meaning of animal diversity? Animal diversity is nothing but all the animals living together on the earth. Okay, it is called as animal diversity. All the animals living together is called as animal diversity. So, to see, it is very simple. Living together is very simple. But it is a very complex relation is there among all the animals. Okay, so here simply animal diversity means all the animals living together. Okay, so here based on vertebral column or backbone, as said madam, based on vertebral column, we classify all the animals into two groups, vertebrates and invertebrates. Okay, vertebrates means the animals which have vertebral column or backbone or spine is called as vertebrate. The animals which have no vertebral column is called as invertebrate. So based on vertebral column, we classified animals into invertebrate and vertebrate. Okay, so under animal diversity 1, so in our textbooks, there are 2 animal diversities are there. For our compatibility, under animal diversity 1, we will learn invertebrate phyla. Under animal diversity 2, we will learn vertebrate phyla. Okay, right now we are going to start invertebrate phyla. So, in invertebrate phyla, under an invertebrate phyla, invertebrates, we are going to study following phyla like Porifera, Cnidaria, Ctenophora, Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca, Echinodermata, and Hemichordata. These 10 phyla we are studying under Animal Diversity 1. Okay, so as you are newcomers for intermediate education, so to make interest on zoology, I would like to give little and brief introduction. So Earth was formed about 4.5 billion years ago. Our Earth, which we are living on Earth, our Earth is formed 4.5 billion years ago, and life was started about 3.5 billion years ago. There are around 8.7 million species are there on Earth. To study all the species systematically, various disciplines were gradually developed. Okay, and zoology is the one of the disciplines to study about animals. To study about animals, there is a special discipline designed for us, that is zoology. As per R.H. Whittaker, R.H. Whittaker is a taxonomist. R.H. Whittaker as per R.H. Whittaker classification, all the living things on this earth is classified into 5 kingdoms. Okay, so to study systematically all the animals, the classifications is necessary. So to classify animals, there are few hierarchical levels are there in classification. So see, these diagrams tell you that what are the levels in classification. The bottom level is called species and the highest level is called life. Okay, so life is classified into domain. Domain is classified into 5 kingdoms. As per R.H. Whittaker, 5 kingdoms are there. All the animals are classified into 5 kingdoms. So what are those 5 kingdoms? You can see Monera, Protista, Fungi, Plantae, and Animalia. Okay, so kingdom Animalia, the last kingdom is Animalia. The kingdom Animalia is divided into 2 subkingdoms. Based on what? Based on tissues. Based on tissues, Animalia kingdom is classified into 2 subkingdoms like Parazoa and Eumetazoa. See here, Parazoa and Eumetazoa. This Animalia is also called as Metazoa. This Animalia is also called as what? Metazoa. So when we see the Parazoa, Parazoa has only one phylum, that is called Porifera. Rest of all the phylums comes under Eumetazoa. Okay, so Eumetazoa starts from Cnidaria and with Chordata. So our first phylum is Porifera. See, what are the general characters of Porifera? So Porifera means pore-bearing animals. So see, in this diagram, I have presented a poriferan animal to you. When we see this animal, under their body surface, we can find tiny pores. As it has these kind of tiny pores, these animals are called as poriferans. Poriferans means pore-bearing animals. Pore is nothing but a small or tiny hole. The term porifera coined by Robert Grant. For the first time, this term used by Robert Grant. Their animal nature was also developed by Robert Grant. Earlier, most of us believed that poriferans are plants, because most of the poriferans cannot move and they look like plants. So for the first time, their animal nature established by Robert Grant. Poriferans also called as sponges, as they look like sponges. These are also called as sponges. They are generally marine, but few are freshwater sponges. Usually we can see in marine water. Marine water means salt water. Where we can find salt water? In seas and oceans. So they are live in seas and oceans usually. But a few sponges can live in freshwater also. So when we see their symmetry, usually they have radial symmetry. Radial symmetry, what is radial symmetry? You have studied in previous lesson. Radial symmetry means when we cut through any plane, the individual comes into two equal halves. That type of symmetry is called as radial symmetry. But in few animals, we cannot find any symmetry. Those animals are called as asymmetrical animals. See here, I have presented few images for you. In first two images, there are few types of sponges are there. These sponges are marine water sponges. This green color sponge is a freshwater sponge. So these are the sponges diagrams which are given in our textbook. First two sponges, Cycon and Euspongia, are marine water sponges, and the third one, Spongilla, is a freshwater sponge. These are primitive and multicellular and sessile. These are primitive, multicellular and sessile. Primitive means first animals, oldest animals. Multicellular means made up of more than one cells. Sessile means stay at one place, fixed at one place. Okay, so these animals, oldest animals, multicellular animals, are fixed at one place, cannot move. And these animals have cellular level of organizations. Their body organizations permitted at cell level only. Therefore, their body organizations is called as cellular level of organization. The body wall composed of two layers. Outer layer is called pinacoderm and inner layer is called choanoderm. These two layers separated by gelatinous matrix is called mesohyl. In between these two layers, pinacoderm and choanoderm, we can find gelatinous structure or gelatinous matrix is called mesohyl. Sponges have water transport system or canal system. This is important to remember. Water transport system or canal system. So on their body surface, we can find a tiny pores which allows the water inside their body. Those pores are called ostia. Through the ostia, water constantly enters in their body. And the water comes out from a larger opening is called osculum. Okay, these two pores are important, ostia and osculum. Water enters through ostia and comes out through osculum. The transport of water helps in gathering food, exchange of gases, and removal of waste. As it is performing key and important function, this is important for us. The water transport system is important for us. What is the duty of water transport system? It gathers the food, exchange the gases, and removal waste from their body. See, this is water transport. So water transport system, to understand this diagram helps you. Okay, through this tiny pore, water enters and through osculum, water comes out. Nutrition is holozoic nutrition. Holozoic nutrition means it is a type of heterotropic nutrition. Holozoic nutrition is a type of heterotropic nutrition, and the digestion is intracellular digestion. If the digestion takes place inside the cell, that is called as intracellular digestion. See, in this diagram, there is a cell membrane is there. If the digestion takes place inside the cell membrane, then that is called intracellular digestion. If the digestions take place outside the cell membrane, then that is called extracellular digestion. And the reserve food stored in thisocytes. There are specialized sites are there. Sites means cells are there. Those cells are called thisocytes. The main function of these thisocytes are reserve the food. So visually, when you get excess food, that excess food should be stored in thisocytes. And nerve cells and sensory cells are absent in these animals. Tissues not formed, and nerve cells and sensory cells are absent. As nerve and sensory cells are absent, the coordination among body parts is much slow. The coordination should be much slow among body parts because lack of nerve cells and sensory cells. The skeleton made up of calcareous cells. The calcareous should be made up of calcium carbonate, therefore these are called as calcareous cells. The skeleton should be made up of calcareous cells. See, in these diagrams, I have shown calcareous cells. How are calcareous cells? And sexes are not separate. See, in human beings, male and female are separate. But in sponges, sexes are not separate. A single individual can act both male and female characters. It has male parts and female part. Therefore, sexes are not separate. Which animal sexes are not separate? Those are called as bisexual or monoecious. As it is monoecious, eggs and sperms are produced by the same individual. Sponges are reproduced asexually by fragmentation, budding, etc. and sexually by formation of gametes. So two types of sexual reproductions we can see in sponges: sexual reproduction and asexual reproduction. The asexual reproduction usually happens through fragmentation and budding. Fragmentation and budding are common reproductive methods, common asexual reproductive methods in sponges. And the sexually gametes fuse each other, and that is called sexual method. It can produce gametes, male gamete and female gamete. Those male and female gametes produce the fuse inside the body, then it can reproduce. So sexual reproduction is there and asexual reproduction there in these primitive animals. Fertilization is internal. After fusion the male and female gametes, fertilization occurs inside the body. Therefore, fertilization is internal. The cleavage is holoblastic cleavage is there. So here, this is a type of holoblastic cleavage. The egg completely divides into blastomere. That is called holoblastic cleavage. And development is indirect development. So after fertilization, the zygote directly cannot develop into same as adult. In between zygote and adult, there should be intermediate stages there, that is called larval stage. If the larva is present, then the development is called as indirect development. So already you have studied in your lower classes. So usually in every pond around us, we can find tadpole larvas, tadpole larva of frog. The tadpole larva is frog, it looks like fish, not it looks like frog. So gradually tail disappears and finally it looks like say mother or father, look like adult. But in intermediate stage, it cannot look like adult or their parent. That is called larva. So few larvas are there for sponges. Those are parenchyma, trichymella, amphiblastula. These are few larvae for sponges. See, my dear children, all these larvae very important for your competitive examination. So try to remember parenchyma, trichymella, amphiblastula. Larva is morphologically different from the adult. Morphologically, to see, larva should be different from adult. I told you tadpole larva how appears. So power of regeneration is well developed. What is the power of regeneration? What does it mean? When an organ ampute itself or something else by something else, then that organ can regenerate, regrow. That capability is called as regeneration. So power of regeneration is well developed in these animals. So few examples are there for sponges. Those are cycon, euspongia, spongilla. These are few examples for sponges. Classification. So when we see the classification of porifera, the porifera is classified into three classes, namely Calcarea, Exactinellida, and Demospongia. The porifera is classified based on different characters. There are three classes are there: Calcarea, Exactinellida, and Demospongia. Calcarea. Let us see Calcarea at first. Calcarea animals live in shallow marine water. Where they can live? They can live in shallow marine water. Shallow marine water means where the water level depth is less. There these animals can live. And these animals are solitary or colonial. Solitary means living alone. They can live alone. Around them we cannot find other animals. Colonial means by living together. Okay, so few animals can live alone and few can live together. The body structure is simple. So anatomically, when we see, their body structure is very simple to understand. And spicules are calcareous. Okay, their skeleton is made up of spicules. These spicules are calcareous spicules. These spicules are called as calcareous. Why these spicules are calcareous? Because these spicules are made up of calcium carbonate. As these spicules made up of calcium carbonate, these spicules are called as calcareous. Calcarea example. See, calcarea example, cycon, leucosolenia, and grantia. So here, cycon is there and leucosolenia is there. See how these are. Hexactinellida. The second class is Hexactinellida. These animals living deeper parts of the sea. So in first class, those animals, calcarean animals live in shallow marine water. Hexactinellida animals live in deeper parts of the sea. They are solitary, live alone. They cannot form any colony. They can live alone. Spicules are 6-ray. From every spicule arise 6 rays. Therefore, this spicule is called as hexactinal. And spicules are siliceous. Siliceous means the spicules made up of silicon dioxide. As it is made up of silicon dioxide, the spicule is called as siliceous spicule. Silicon dioxide chemical formula, you know, SiO2. This is also called glass. Hexactinellida example. Euplectella. See, this is Euplectella. See how it is beautiful. This Euplectella is also called as Venus flower basket. This is commonly called as Venus flower basket. As it is appear as a basket, this is called as Venus flower basket. And this is also called as marriage gift of Japan. Usually we can present bouquet in marriages, flower bouquet. In Japan, most of the places, they can present Euplectella in marriage ceremonies. Therefore, this is called as marriage gift of Japan. The second one is Hyalonema. The Hyalonema common name is glass rope sponge. See, this is Hyalonema. In this diagram, you are watching Hyalonema. So, it is look like a rope. Which rope? The rope which made up of a glass. Therefore, this is look like a glass rope. Therefore, this is called as glass rope sponge. Hyalonema is also called as glass rope sponge. The third class is demospongy. In this class, both marine and freshwater sponges are included. So in first two classes, exclusively marine sponges are there. But in this class, freshwater sponges are also included. See the first diagram, the first image, this is a freshwater sponge. They are colonial. They cannot live alone. They are colonial. Skeleton consists of siliceous spicules or spongin fibers, or both the combination of siliceous spicules and spongin fibers. In some sponges, skeletal structure may be absent. There is no guarantee. In some species, skeletal structure may be absent. See demospongia example. Spongilla, freshwater sponge. Euspongia, this is also called as bath sponge. Chalena, this is called dead man's finger. Spongilla is called as freshwater sponge because we can find it in freshwater, therefore it is called as freshwater sponge. Euspongia, as this sponge used as a used to scrub the skin, therefore this is called bath sponge. Chalena, this is also called dead man's finger. When the human beings die, then his finger look like a freezed finger. How the dead man's fingers appear, this chelina appears same as. Therefore, this is called dead man's figure. Okay, that's about phylum Porifera. Right now, we are going to study about Cnidaria. The first phylum Porifera is over. The second phylum, we are entering into second phylum, that is called Cnidaria. Cnidaria is a new name. This is also called Coelenterata. Coelenterata is a old name. This phylum has two names. When Coelenterata gives, please do not confuse. Coelenterata is also called as Cnidaria. Cnidaria is also called as Coelenterata. But what is the specialty of these animals? In these animals, tissues form. These animals are first tissue formed animals. Tissue started from these animals. This is the significance of these animals. And in these animals, usually we can find radial body plan, radial symmetry. Let us see what are the common characters of these animals. Cnidarians previously called as coelenterats. As I told, previously these are called as coelenterats, and these are aquatic. Exclusively we can find only in water. We cannot find on land. And mostly marine. Most of the animals live in marine water only. Solitary are, these are solitary or colonial. Living alone or living together. Sessile are free swimming. Sessile means fixing at a particular place. That is called sessile. Sessile mode of life. Like plants. They cannot move from one place to other. They can fix at one place and they can stay there only. It cannot move for anything, for food or shelter or for anything. When food comes to it, then it can catch. But it cannot move. So few are sessile and few are freely swimming animals. When we see the symmetry, their body is radially symmetrical. But try to remember, children, sea anemone. Sea anemone is there. It's a well-known animal for us. The sea anemone's symmetry is biradially symmetrical. Usually their body symmetry is radial symmetry, but sea anemone symmetry is biradially symmetrical. Body and tentacles covered by stinging cells. This is a special property of Cnidaria. Having stinging cells is very important and very unique property of Cnidarians. The name of those stinging cells are cnidoblast or cnidocytes. These are also called cnidoblast or cnidocytes. Okay, and the word Cnidaria derived from cnidoblast cells. We put this phylum name as Cnidaria. Why we put Cnidaria? Because as it is having cnidoblast cells only. Okay, cnidoblast are cnidocytes. As it having cnidoblast and cnidocytes, these animals are called as cnidarians. So what is the use of cnidoblast cells? You can see. Cnidoblasts are used for anchorage, defense and capture the prey. Cnidoblast cells used for anchorage. Anchorage means hold the surface. To do not flush out, it can hold the surface with the help of cnidoblast cells. When its enemy attack, then it can use cnidoblast cell to escape. So these cnidoblast cells used in defense also and capture of prey. It's also used for capture of prey or food. Okay. So see in this diagrams you can see cnidoblast cells. These cnidoblast cells perform the following functions. Cnidarians are the first metazoans to exhibit tissue level of organization. And these are the first metazoans to exhibit the diploblastic nature. These are not first, I think. It has diploblastic nature. Diploblastic nature means it has two layers, two germinal layers, ectoderm and endoderm. Okay, see in these diagrams you can see ectoderm and endoderm. Here in these diagrams you are also watching mesoderm, but mesoderm is absent in these animals. Try to remember. We can find only two layers, two germinal layers like ectoderm and endoderm. Mesoderm is completely absent in these animals. They have central gastrovascular cavity or cylindron. See, here in these diagrams, you can see the middle of this pod is called cylindron. Okay. As it has cylindron, this is also called cylinderator. The name cylinderator derived from cylindron. This cylindron serves two important functions like digestive functions and circulatory functions. And cnidarians have single opening that is called mouth. Usually, all the developed organisms have two important openings like mouth and anus. But these animals have only one opening, that is mouth. Okay, mouth serve for both ingestion and egestion. What is ingestion? The process of taking food is called as ingestion. The process of taking food, water inside the body, then after it can digest and assimilate, that process is called as ingestion. Egestion means after digest the food, the undigested food comes out from the mouth, that is called egestion. So mouth serve for both ingestion and egestion. These two important functions served by mouth only. As anus is absent in these animals. Digestion is in both extracellular and intracellular. Already I told you, if the digestion occurs inside the cell membrane, in the cell inside the cell membrane, if the digestion occurs, then that is called intracellular digestion. If the digestion takes place outside the cell membrane, that is called extracellular digestion. In these cnidarians, extracellular digestion takes place at cylindron. And intracellular digestion takes place in nutritive muscular cells of endoderm. In endoderm, we can find especially cells are called nutritive muscular cells. There we can find intracellular digestion. So, however, cnidarians, digestion take place in two methods like extracellular and intracellular. And nervous system is primitive and formed by non-polarized nerve cells called nerve net. Okay, so nerve net means interconnected neurons are there. Interconnected neurons. The interconnected neurons are called as nerve net. Nervous system is primitive. Primitive means oldest and firstly formed. Nervous system is primitive and formed by non-polarized nerve cells. These are formed by non-polarized nerve cells. These nerve cells is also called as neurons. Due to fusion of these neurons, the nerve net forms, but brain cannot form. So this is about nerve net. This is about the nervous system of these animals. And sensory structures such as statocysts occur in medicide forms. So usually two types of body forms are there, like polyp and medusa. This is the specialty of these animals. So when it is in medicide form, we can see some statocysts. These are the sensory cells. So when it is in medicide form, we can find statocysts. What are statocysts? Statocysts are sensory structure or sensory cells. So few cnidarians can form corals. These coral forming cnidarians have an exoskeleton. This exoskeleton is composed of calcium carbonate. So examination point of view, this is very important. The coral forming cnidarians is made up of calcium carbonate. The exoskeleton is made up of calcium carbonate. Cnidarian shows two basic body forms. As I told you, the cnidarians can show two basic body forms like polyp and medusa. So we will see what are the differences between these two. Polyp and medusa. See, this is polyp and this is medusa. When it is in polyp body form, then it is sessile. Polyps are sessile. Medusae are mobile. They cannot move, but medusae can move from one place to other. And polyp are tubular or cylindrical in shape. This is cylindrical in shape, tubular in shape. This is bell or umbrella shape. This body form You can see this is umbrella shaped body form. And the water faces upwards in these animals. When it's in polyp stage. So the water faces downwards in Medusa. The sense organs are absent. Here sense organs are present. Okay, photoreceptor and tentacles are present in Medusa form. The best example for the polyp, Hydra. Freshwater polyp it is. Adamsia, sea animal. This is called, Adamsia is called as sea animal. Hydra and Adamsia, best example for polyp, and jellyfish is best example for Medusa. This is important for 4-mark question also in our IB. See, this is Medusa, this is polyp. Okay, I think this diagram, this diagram given in our textbook. Cnidarians reproduction. When we see the reproduction, sexual and asexual reproductions are there. Cnidarians, common asexual reproduction is budding, and sexual reproduction is called syngamy. See, here in this diagram, so you can see budding. This is an individual hydra. From this hydra, a bird, a small bird arising. This small bird grow up. Finally, this small bird grow up and detach itself, can grow up as individual. This type of asexual reproduction is called budding. Okay, and another type is syngamy. This is sexual reproduction. Budding is a asexual reproduction, and syngamy is a sexual reproduction. When you see the syngamy, two cells are their nuclei when they fuse. Okay, by the fusing of the two individual cells are their nuclei, it can produce many young ones. That is called syngamy. This is a sexual reproduction. And usually development is indirect development. When indirect development comes, there should be a larva is present. The larva is called planula larva. It's a freely swimming larva. Planula larva is there in cnidarians. See in this diagram, there is a small, tiny larva is there. This is called planula larva. A few examples for cnidaria: Phycelia, Adamsia, Pennatula, Rhizostoma, Menandrina. All these are example for cnidaria phylum. See in these diagrams how beautiful these cnidarians are. When we see the classification of cnidaria, the cnidaria is classified into 3 classes: Hydrozoa, Scyphozoa, and Anthozoa. These 3 are 3 classes of cnidaria phylum. Let us see Hydrozoa first. They may be solitary or colonial. You know what is solitary and what is colonial. And sedentary. Live at the bottom of the seas or bottom of the oceans or bottom of the freshwater, lakes or streams or something else. And we can see metagenesis in these animals. What is metagenesis? Sexual and asexual forms alternative with each other. Sexual form and asexual form alternative with each other. This is one of the important characters of cnidarians. This is called metagenesis. Sexual and asexual method, when alternative comes, then that is called metagenesis. The mesoglea is non-cellular in these animals. So presence of mesoglea is common in these animals. This mesoglea is non-cellular. We cannot find any cells in this mesoglea. And pneudoblasts occur only in ectoderm. This is a diploblastic animal. Endoderm and ectoderm we can see. The cnidoblast occurs only in ectoderms only. And germ cells are derived from ectoderm only. The best example for Hydrozoa is Hydra. What is Hydra? Hydra is a freshwater polyp. Next is Scyphozoa. Class 2, Scyphozoa. Scyphozoans are commonly referred to as jellyfishes. All the jellyfishes included in Scyphozoa. They possess a large quantity of jelly-like mesoglea. Okay, they possess jelly-like mesoglea. They are solitary and medusoidal forms. Free swimming form is there and staying at one place, that is called solitary form is there. Mouth is surrounded by 4 oral arms. Around the mouth, we can find 4 oral arms. Cylindron is divided into 4 chambers and canals. That cylindron is divided into 4 chambers or 4 rooms. And mesoglea contains amoebocytes. When we studied the first class, there is no cells, that mesoglea is non-cellular, but in this Scyphozoa, mesoglea contains amoebocytes. And pneumatoblast occur in ectoderm and endoderm. The pneumoblast should be present in both layers, ectoderm and endoderm. And germ cells derived from ectoderm only. The best example for this class, best examples are Aurelia and Rhizostoma. Aurelia is also called jellyfish or moonfish or moon jelly. And Rhizostoma. Aurelia and Rhizostoma, these two are best example for Scyphozoa. The third one is Anthozoa. Anthozoa is also called as Actinozoa. They are commonly referred to as sea anemones. Sea anemones included in this Anthozoa. Okay. Hydrozoa, Scyphozoa and Anthozoa. Hydra included in Hydrozoa. Jellyfishes are included in Scyphozoa. Sea anemones are included in Anthozoa. Try to remember. They are commonly referred to as sea anemones. Anthozoans are commonly referred to as sea anemones. They are sedentary and polypid form. Their lifestyle is sedentary and they are in polypid in form. Cylindron is divided into several compartments. In previous class, 4 compartments we have seen. But here, several compartments. Cylindron is divided into several compartments. And mesoglea contains connective tissue. Okay, mesoglea contains what? Connective tissue. And cnidoblasts occur in both ectoderm and endoderm. You can find cnidoblast cells both in ectoderm and endoderm. Germ cells are derived from endoderm only. These are the best example for the class Anthozoa, Adamsia, Gorgonia, Pennatula, Corallium rubrum. These 4 are best example for Anthozoa, and their common name very, very important for examination point of view. All the common names of these 2 phylums very, very important for examination point of view. Most of the time asked in competitive exams also. So try to remember. Adamsia common name is sea anemone, Gorgonia common name is sea fan, Pennatula common name is sea fin, Corallium rubrum common name is precious redstone. Okay, so this is about Anthozoa. This coral is very important, Corallium rubrum, precious redstone coral it is. Okay, this is about Anthozoa. And so once you can see a recap. So the 2 phylums over. When we see the Porifera, in Porifera you have what you have observed, these are the first multicellular animals. Tissues could not form in these animals. And cnidarians have different type of cells. And All the cells have their own functions, and ostia and osculum you have find in these animals. Through the ostia, water settles inside their body. Through the osculum, water goes out from their body. And the canal systems you have find in these animals. Okay, that canal system plays important role to gathering the food, exchange the gases, and removal of waste. And holozoic nutrition is there, intracellular digestion is there. Histiocyte reserve the food. Okay, so these poriferums are classified into 3 classes: Calcarea, Hexactinellida, Demospongia, like that. So Calcarea examples, try to remember: Cycon, Leucoselenium, and Grantia. Hexactinellida example, try to remember: the Calcarean spicules made up of calcium carbonate. Hexactinellida animals spicules made up of silicon dioxide. Demospongia examples: Spongilla, this is called freshwater sponge. Euspongia is called bath sponge. Chalina is called dead man's fingers. All these important for examination point of view. So try to remember. And when we see nidaria, that tissues for the first time formed in these animals, that is an important character. And why these are called as nidaria, and why these are called as Coelenterata, this is also important. And having stinging cell is a very important characters, one of the very important characters of these animals. So try to remember pneumoblast cells and pneumocytes and their functions also, anchorage, defense, and captures, like this. So having coelentron is also one of the important character of these animals. Digestion should be extra and intracellular. There is a nerve net is there. And having statocyst, examination, MCET examination point of view, this is important, try to remember. And when it is in medullary form, we can find statocyst. This is a sensory cell. Statocysts are sensory cells. And coral forming cnidarians made up of calcium carbonate. This is also important for examination point of view. And MCET point, the IP point of view, the differences between polyp and medusa is important for us. And common characters of anthojuvans, very important for Phomacuchans. Silent features of anthojuvans is very important for Phomacuchans IP point of view. These are Aurelia and Rhizostoma example for Scyphozoa class. Adamsia, Gargonia, Pennatula, these are example for Anthojova. All these try to remember. Thank you very much. The session is over. And thank you, students. And thank you so much, one and all. Thank you, madam. It's over, madam.
Thank you very much, Shankar sir, for this interesting explanation of the porifera and nidaria phyla of the animal kingdom. I'm sure all students must have gained a clear understanding of animal classification. The part about sponge regeneration, where a complete adult can grow from new fragments, which indeed very fascinating. When you grind a sponge, each fragment can develop into a new sponge. It is relevant to note that one of the world's great natural wonders of the world, the Great Barrier Reef of Australia, is made of colonies of coral polyps, which are cnidarians. And there is an additional thing that I would like to point out. Sometimes the kingdom Monera is divided into 2 separate kingdoms, Archaebacteria and Eubacteria. Therefore, some sources say that there are 6 kingdoms, and but in our textbook, it is 5 kingdoms are considered. So there should be no confusion regarding this. And I request all the students to read the textbook chapter thoroughly, watch this video again and again, and prepare the notes. Thank you, sir, for the presentation. And thanks to all the students for attending. I hope all students find these sessions useful and continue to participate in large numbers in the future as well. And students may please subscribe to the Model School Telangana Junior Intermediate YouTube channel to stay updated with the latest subject content. Thank you once again, sir. Shankar sir. Thank you.
Thank you so much, madam.